Let's go over some of the basics and some of the basic concepts and some of the basic structure that we see with xylem vessels. So, when we are going to talk about it, we first have to understand that xylem vessels have a few characteristics that are quite unique or quite unusual. We don't see these characteristics with a lot of other tissues, right? What are they? What am I talking about? So, first of all, your xylem vessels are hollow tubes. That's what they are. They're hollow tubes. They are continuous. It's a continuous tube. Okay. It's a continuous tube. There's no separation between the cells. And this is important because you guys are going to see how this makes it different from phloem. So it's a continuous hollow tube. It's a continuous tube, and it is actually made from cells. It's made from plant cells that are joined from one end to another. So just imagine that you have taken small bricks and you have put them one on top of the other, on top of the other, on top of the other to make a tower, to make a tower-like structure, right? Similarly, xylem vessels are made up of cells that are joined from one end to another. So xylem vessels that are stacked on top of each other, right? The cells are basically dead. They're not live cells, which means that they are not going to give you any of the features that live cells give you. They're not going to have any cytoplasm. They're not going to have any nucleus, no mitochondria, nothing like that at all. No transport is going to be taking place. The cell will not be able to function in any way at all. So why is it that xylem vessel is made up of dead cells? We will see in just a bit. Why is that so important? But the cells that make up xylem vessels are dead. And the cell wall of the xylem vessel, the cell wall of the xylem cells is lignified. What is lignified? What do you mean by lignified? Lignified is the term for the deposition of lignin. Lignin is a material that provides extra strength and extra support to the extra strength and extra support to the cell wall. So when we are going to go ahead and talk about it, let's now take a, so this is the basic concept of what xylem tissue is. And you guys might have even studied this in your IGCSE, but we need to understand this concept in further detail, right? When we are going to talk about it, now let's go over the structure of our xylem vessel. Okay, what is a xylem vessel? What does it actually look like? So our xylem vessel, uh, let's start from the initial scenario. Let's say that this is a young plant. The xylem vessel is just being made. Okay, it's not made yet. It's not ready yet. So it's just being made. So xylem vessels are made up of many elongated cells. Xylem vessels are made up of many elongated cells. The name of these cells is, we call them vessel elements. That's the name of the cells. So the name of the xylem cells is, what is the name of the xylem cells? The name of the xylem cells is that we call them vessel elements. Now, when we are going to, let's start with the initial situation, right? Initial situation is that right now the xylem vessels, the plant is still growing. The xylem vessels are not ready yet. So each vessel element means that each cell, okay? Each vessel element, or if I write it down easier, each cell of the xylem vessel, okay? So each cell begins its life as a normal plant cell. Each cell begins its life as a normal plant cell, meaning that it is going to have a nucleus, it's going to have cytoplasm, it's going to have chloroplast, it's going to have everything. It's going to be like a normal functioning cell of the plant. That's what it's going to look like. Now, what is going to happen that in the cell wall of these cells, lignin is slowly deposited. Slowly, little by little, I will start depositing lignin on the cell wall of these normal plant cells. What's going to happen? What happens is this, that lignin is impermeable to water, meaning that it does not allow water to actually pass through. It's impermeable to water. It doesn't allow water to pass through, right? So lignin is impermeable to water and extremely strong. How strong? You guys know how your trees have wood, right? That wood is lignin. That's what lignin is. So now what is going to happen as more and more lignin deposits, as more and more lignin is going to deposit, what is going to happen? As more and more lignin is going to deposit, water is unable to enter the plant cell because lignin is impermeable to water, doesn't allow any water to travel through. So water cannot enter into the plant cell. As a result, the vessel elements or the vessel elements are what? The cells. That's the name of the cells, right? Start to die. That's why it is made up of completely dead cells because of deposition of lignin, which is good for us because it makes the walls of the xylem vessel strong. So water is unable to enter the plant cell, and as a cell, the vessel elements are going to start to die, right? Now, this continues until all the cell contents are gone, until the cell is dead. When the cell is completely dead, all the contents, all the cytoplasm, nucleus, mitochondria, chloroplast, ribosomes, everything, it's all gone because the cell is now dead. No water was able to pass through, and the cell has slowly died, leaving behind a hollow tube. Because of the fact that the cells are all dead, there is no cytoplasmic or no cell content. There is no cell content. There is no cytoplasm, nothing. So what happens because of this? What happens because of this is that there is no obstruction in the pathway of water. The tube of xylem vessel, the tube is empty because the cells are all dead. There's nothing in the way. It's a hollow tube. It's an empty tube, right? So that's why lignin is so good, because not only is it actually making the wall stronger so that the xylem vessel can provide support to the plant, but it is also making it impermeable so water is not able to leave or enter, which makes the cell die. And as the cells will die, all of the organelles, cytoplasm, everything within the cell is, of course, going to be lost, leaving behind a hollow tube. A hollow tube that is impermeable to water. So we have a hollow tube, and that hollow tube is impermeable to water, meaning that this is the ideal thing that we can have for transport of water, because not only is it completely hollow with no obstructions in the way, but it is also impermeable to water, meaning that as water is going to travel up the xylem vessel, it's not going to just move out. It's not just going to spread out of the xylem vessel. No, it will stay within that vessel. Why? Because there is lignin all around it that makes it impermeable. So water is able to travel through the xylem vessels without disruptions, without anything stopping it, right? Now, you can ask me, Miss, if xylem vessels are surrounded by lignin, If xylem vessels are impermeable to water, then how does the water actually leave the xylem vessel and go to the leaf or the fruits or the flowers, wherever it has to go? How does it actually do that? So it does that with the help of small openings in the lignin, in the xylem vessel wall. There are small openings. Those small openings are known as pits. So the pits allow to move from the xylem vessel to the leaf, for example, right? So if the water has to move out from the xylem vessel into the leaf, they will move out with the help of those pits. Now, what are pits? How are pits made? How are the pits formed? So when we're going to talk about it, if you guys remember, I had taught you guys about, when we did the topic of cell biology, I had told you guys that the plant cells are connected to each other by plasmodesmata, right? Plasmodesmata is this sort of connection. It's sort of this channel that connects one plant cell to another, right? And it has um, cytoplasmic extensions inside it. So inside the plasmodesmata, we have extensions of the cytoplasm. The cytoplasm for both the cell will extend into the plasmodesmata. And that is what allows one plant cell to communicate or talk with another plant cell. So what happens? No lignin is deposited around the plasmodesmata. So the area of plasmodesmata remains non-lignified. There is no lignin deposited in the area around the plasmodesmata. Since there is no lignin deposited around the plasmodesmata, so that particular area will remain normal, meaning that it will be permeable to water. So in the area of plasmodesmata, lignin is not deposited, right? Since the lignin is not deposited, right? What is going to happen? That this makes small gaps in the xylem wall. So because if the entire xylem wall is lignin, except for the area of the plasmodesmata. In that area, there's no lignin, right? So there's no lignin, but that doesn't mean that there's nothing there at all. We still have the cell wall. The cell wall is still there, okay? The cell wall of the plant is still there. The cellular cell wall is still there. It's just that the lignin is not going to be present. Okay? So this area is known as pits. So this allows the water to actually go ahead and go from one area to another. So it can go from the xylem vessel to the surrounding leaf, to the surrounding tissue, to the surrounding whatever area actually requires that. Okay, so this was about one single cell. So what's going to happen? Multiple vessel elements um will arrange or will stack end to end to make one continuous hollow tube, right? Now, what is going to happen that generally speaking, between the cells, we will also have the cell wall, right? Because the cell wall surrounds everywhere. The cell wall will be on top of the cell, on the left of the cell, right of the cell, below the cell, it's going to be everywhere. Okay, so the sides of the cell are all lignified. What about the end areas, right? Meaning, what do I mean by that? if this is my cell that I'm stacking on top of each other like this, right? So this outside area you guys lignified, fine. This was lignified, this was lignified. What about this? What about the part of the plant cells that is arranged on top of each other? What happens to that, right? Let's take a look. What happens to this? So what happens to that is that the cell wall found between two vessel elements, the cell wall found between two vessel elements as they are placed on top of each other is called the end plate. Now, for it to be a continuous tube, we also need to get rid of the end plate itself, right? So what is going to happen? The end plate or the end walls will also break down. They also need to break down, okay? The end walls will also break down, providing a continuous pathway to the water. The end walls in the middle are also going to break down, providing a continuous pathway in the water. And this is how the xylem vessels are actually going to be working, right? This is how this is the basic structure of your xylem vessel. So, right? So this is the your structure of your xylem tissue. Now, xylem tissue is not just made up of vessel element. Okay. Xylem vessels, when we're going to go ahead and talk about it, xylem vessels include four structures. Vessel elements, which I've already taught you guys. Then we have something known as tracheids, which I will tell you about in just a bit. Then we have a certain fiber that I mentioned to you guys previously as well, that we're going to study a fiber. That fiber is known as sclerenchyma fiber. And we have, of course, parenchyma cells as well, which you guys have already studied with me. Now, when we are going to go ahead and talk about it, so first of all, let's go over sclerenchyma fibers. What are sclerenchyma fibers? Sclerenchyma fibers are elongated cells, okay? Are elongated cells. They are elongated cells with lignified walls that help to support the plant. Just like vessel elements, they are also dead cells with no living contents, right? So, for example, if we go ahead and we take a look of the xylem vessel, what is the xylem vessel actually going to look like? It's going to look like this. So this is my xylem cell. This is my xylem vessel. Okay. Now, in my xylem vessels, I'm going to have pits, which I'll show you. This is our lignified cell wall, and then we have a hollow lumen at the center. Then let's say I have another xylem vessel over here. Now, over here, I will be seeing these pits. This is your pits. You see how it's a connection that is between the two xylem vessels? Similarly, that connection can happen between your xylem vessel and your plant cell, your xylem vessel and the fruits, your xylem vessels and the flowers. So you can see this kind of structure throughout. So this is going to be my xylem vessels. All of these vessels are going to be able to openly communicate with each other. And when I say communicate, I simply mean the transport of water. In terms of transport of water, there are going to be pits present that are going to allow the water to leave the xylem vessel. So the water can only leave the xylem vessel through the pits. That's it. Otherwise, it is not able to leave the xylem vessel. It has to stay inside. So this is my structure with the xylem vessels. Now, aside from the xylem vessels, what else do we actually see in our in the xylem tissue? So these were the vessel elements which I told you guys about. Okay, I have drawn all the vessel elements for you guys. Now, aside from vessel elements, we have something that is known as our sclerenchyma. So our sclerenchyma are basically, and I'll draw the a picture for you guys as well, what it actually looks like. But our sclerenchyma is actually these elongated cells that are also lignified. And you can find them around the xylem vessels like this. So they are these elongated cells that are lignified. And you will find them all around your xylem tissue. You're going to, sorry, I'm saying xylem tissue, I mean the vessel elements. All around your vessel elements, you're going to find the the sclerenchyma fibers. The sclerenchyma fibers are elongated cells. They're like really long cells, and they are, generally speaking, also lignified. They're also dead. They also have a hollow lumen, just like the other ones. So after sclerenchyma fibers, we can have our parenchyma. When we talk about our parenchyma, so you and I already discussed what parenchyma was, if you guys remember, right? We had said that parenchyma was what? Parenchyma was these supporting cells that are found, or the packing cells was the term that I taught you guys. These packing cells that are found in the stem, in the root. I taught you everywhere that we have these parenchymal cells. So similarly, we can have these skeletal cells, and then we can have our parenchymal cells. The parenchymal cells, though, are going to be normal cells, meaning that they're going to have a cell wall, they're going to have a nucleus, they're going to have, your vacuoles. So this altogether makes the structure of these are all my parenchymal cells that I'm drawing for you guys. So cytoplasm, vacuole, nucleus, everything will be seen in the parenchymal cells. So they are the packing cells. They are basically there to provide strength and support. That's why we have these packing cells. So this altogether would, make your tissue of the xylem cells. Now, when we are talking about our, this xylem tissue, so I mentioned to you guys that we will have something known as tracheids as well, tracheids. So what are tracheids then? Tracheids, generally speaking, you guys don't have to know in a lot of detail. The reason you don't have to know in a lot of detail what tracheids are is because tracheids are a feature that is generally seen in non-flowering plants. Okay, flowering plants mostly just have xylem vessels. They tend to don't have any tracheids, right? Your non-flowering plants are the ones that generally have tracheids present. So the difference that you guys just can understand is that your tracheids are generally speaking, they are smaller, tracheids are narrower than xylem cells. They tend to have a bit of, a tapered um, end. They tend to have a bit of a tapered end as compared to your xylem vessel cells, your vessel elements. So they pretty much in terms of function, they're quite similar. They have the same function, which is transport of water. The only major difference that comes up is that where you're going to find them. So very little tracheids, almost none, are found in flowering plants. But a lot of the tracheids can be found in non-flowering plants. So like I said, the function of this is quite similar. So I told you guys are not expected to know the details of tracheids because like I said, you are only expected to know about flowering plants. The topic of non-flowering plants is not a part of your syllabus, so you guys don't have to worry about. This is my vessel element. But then inside my vessel element, specifically speaking, this is the lumen. This is the lignified cell wall. This over here are my pits. These are my sclerenchyma fibers. These are my parenchyma cells. So all together, this would make the entire structure of xylem vessel. So xylem vessel is not just these hollow tubes. It's parenchyma cells, it's the sclerenchyma fibers. If you have non-flowering plants, then you will even have tracheids. Tracheids are just like your vessel elements, just narrower. Okay? Pretty much quite similar to what we have already covered. So we are going to have, so this is what your structure of xylem vessel actually is going to look like. So, so xylem vessels, as you can see, consist of a lot of different structures. It's not just the hollow tubes, it's a lot of different structures as well. So, when we are going to go ahead and we are going to talk about it, now that we understand the structure of the xylem vessel, let's talk about how the water is transported. Sorry, guys, one second. how the water is transported from the root to the stem and the leaf, right? How is the water transported from the roof to the stem and the leaf through xylem vessel? So through xylem vessel, how is the water actually going from the root to the stems and the leaves of the plant? So when we are going to go ahead and we are going to be talking about it, first of all, um, as you guys know, that water is going to move from the xylem vessel into the leaf, right? And we studied about how that happens, the symplastic pathway and the epiplastic pathway. So water moves from the xylem vessel into the leaf. We had already studied that. Now, as water will move into the leaf, what will happen? This will reduce the hydrostatic pressure. This will reduce the hydrostatic pressure in the xylem vessels because the water has moved out. The water has gone out from the xylem vessel into the leaf. So the movement of water out, the water has moved out from the xylem vessel into the leaf. So there is less pressure of water in the xylem vessel. There is less hydrostatic pressure in the xylem vessel, right? The hydrostatic pressure at the bottom of the xylem vessel becomes higher than the hydrostatic pressure at the top. And that's the principle I taught you guys as well. When we were talking, I give you the example of a straw. That in a straw, what do? Whenever we apply a suction, we are creating a low pressure area at the top and a high area pressure at the bottom. And that's what causes the fluid to move from an area of high pressure to low pressure. Same thing happens over here. As the water moves from the xylem vessel into the leaf, it creates a hydrostatic pressure, low hydrostatic pressure at the top and high hydrostatic pressure at the bottom. This pressure, so there's a difference in pressure. The top area has less pressure and the bottom area has high pressure. This difference in pressure causes water to move up the xylem vessel, right? In a continuous column. So not only is the water going to move up, but it's going to move up in one continuous column, meaning that in one continuous straw, one continuous row, one continuous stream, no interruptions in the pathway. Now, when we're going to talk about it, now you guys can understand why the xylem vessels need to be lignified. Imagine if you guys have a water bottle, like those plastic water bottles. If you suck the air out of a plastic bottle really hard, what happens? It crushes. Or if you guys are drinking juice from a juice box, and you guys, as you suck from the juice box, right, with the help of a straw, what can happen that if you suck hard enough, you can pull out the air from the juice box and the juice box tends to collapse. It tends to shrink, right? Now, the same thing would happen to xylem vessels because the area at the top has low pressure, area at the high as the bottom of pressure. So it works like a straw. It has like a sucking effect. Now, the xylem vessels should collapse, but the lignification prevents the xylem vessels from collapsing. So it's not just about providing strength to the plant because xylem vessels are also responsible for providing strength. It's not just about providing strength. It's also from preventing the xylem vessels from collapsing because when there would be a pressure difference, it would collapse. They would simply shrink. They would, collapse. But The lignification provides the strength and makes sure that the xylem vessels do not collapse at all. Now, the movement of water up the xylem vessels happens by mass flow. What is mass flow? Mass flow is all the water molecules move together as a body of liquid. For example, like a waterfall, like a river, like a stream, right? They all move together. It's not like a little few of the molecules are going to move, then some of them will move, then some of them will move. It's not like an interrupted movement, it's a continuous movement. That is known as mass flow. The reason the mass flow happens is one, because of the difference in hydrostatic pressure. This is one reason, reason number one, why we see mass flow. Other reasons for mass flow are cohesion and adhesion. What is cohesion and what is adhesion? Cohesion cohesion is the attraction between the water molecules. So the water molecules all have this attraction between them, right? The water molecules are all attracted towards each other. How are they attracted towards each other? This is something I expect you guys to know. How are the water molecules attracted to each other? By hydrogen bonding. They have hydrogen bonding between each other, and that hydrogen bonding between each other attracts the water molecules to each other. This is known as cohesion. Adhesion is the attraction of water molecules to the walls of xylem vessel. So adhesion is the attraction of the water molecules to the walls of the xylem vessels. And this is also due to what? This is also due to hydrogen bonding. Okay. So So this is how the water moves up the xylem vessel. And if I draw the structure of the stem for you guys, so this is what the structure of a stem would look like. I would have my epidermis. Then on the inside, I would have my vascular bundle. Right? And in my vascular bundle, I would have on the outside, I would have my xylem. Sorry, in out the outside, I would have my lignified fibers. Okay. Then I would have my phloem. Then I would have my cambium, and then I would have my xylem vessel. All right. So what is the structure going to look like? Let's go ahead and see. This outside would be my sclerenchyma fibers. This. Then I would have my phloem. Then I would, of course, have cambium, and then I would have my xylem vessel. This altogether is known as my vascular bundle. Then this is my epidermis. This is my cortex. The cortex is made up of parenchyma. It's made up of collenchyma. And finally, we would have the center of the stem. The center of the stem is known as the pith. Okay, so this is the structure of a stem. Now, I have taught you guys how the water actually moves up the xylem vessel. But how does the water, or how are the roots involved in it? Right? Now, when we are going to talk about it, I just mentioned to you guys that the water, when we are going to go ahead and talk about it. So now, what is going to happen? What is going to happen is this, that we need the, I told you guys that the pressure at the bottom is going to be high, right? That's what I told you guys. The pressure at the bottom of the xylem vessel is high, and the hydrostatic pressure at the hydrostatic pressure at the top of the top of the xylem vessel is low, and at the bottom of the xylem vessel is high. Now, it is easy to understand why the hydrostatic pressure at the top of the xylem vessel would be low, because the reason it would be low is because of the fact that the water is moving out of the xylem vessel into the leaf. Since the water is moving out of the xylem vessel into the leaf, so the pressure at the top would be low. But what about the pressure at the bottom? Why is the pressure at the bottom high? We can understand why the pressure at the top is low, but why is the pressure at the bottom actually high? What is happening? Why is it high? It is high because of root pressure. What is root pressure? Root pressure, when you talk about it, plants may increase the pressure difference seen at the top and bottom of the xylem vessel by raising the hydrostatic pressure at the base of the xylem vessels, right? How does it actually do that? So the way it does that is the pressure is raised by actively secreting. Actively secreting means that I'm doing active transport. Actively secreting solutes into water in the xylem vessel in the root. Let me put this in simple terms. I am taking the mineral ions and I'm using active transport to put them in the xylem vessel, right? So what's going to happen? The xylem vessel, the bottom of the xylem vessel, the concentration of mineral ions would increase. If the concentration of mineral ions would increase, the concentration of water or the water potential would decrease. So water would move from the root into the xylem vessel. And as the water would move from the water into the xylem vessels, what is going to happen? The hydrostatic pressure will increase. So what is going to happen? I am doing active secretion. I am using active transport to put the minerals or move the minerals inside the xylem vessel. This reduces the water potential inside the xylem vessel. Hence, the water moves in via osmosis. So the water will move into the xylem vessel, which increases the hydrostatic pressure at the bottom of the vessel. And how does it actually do? How do they do the process of active transport? The way they do the process of active transport is they use pumps. They use pumps that are found across the cell membrane to go ahead and make the water travel into the xylem vessel, right? Okay, so now the water is in the xylem vessel. But the question is, how does the water actually enter the root? Because now we can understand how the water is entering the xylem vessel. But how is the water entering the root? So let's talk about how the water is transported from the root hair into the xylem vessel. Because we can understand why the water is moving up the xylem vessel. That we understand. We understand quite clearly why the water is traveling up the xylem system. We know why the water is moving out of the leaf, transpiration. We know how the water is moving from xylem into the leaf. And now we understand how the water is moving up the leaf. But now we have to talk about how the water moves from the root hair into the xylem vessels, right? So let's draw a diagram. This is, if you guys remember, in the roots, the structure is slightly different. In the roots, the vascular bundle is located towards the center. Remember? So this is my root. And this is my vascular bundle towards the center. And this is my root. And of course, I'm going to have my xylem vessel that's going to be traveling from the center. My xylem vessel. Okay. Now, over here, when I'm going to get to the when I'm going to talk about the root hair cells. So root hair cells are going to form something like this. What do we have present around the vascular bundle? Around the vascular bundle, we have a layer of cells that we call pericycle, which I taught you guys about. So I will have my pericycle. Outside the pericycle, I'm going to have my cortex. Right? Now, outside from the cortex, we are going to have our epidermis. And then from the epidermis, we are going to have extensions of epidermis. The extensions of epidermis are known as the root hair cell. So this is my vascular bundle. This is my xylem vessel. Then I have my pericycle. Then I'm going to have my cortex. Then I'm going to have what? What am I going to have? I'm going to have my epidermis. Then from my epidermis, I'm going to have these hair-like projections. These hair-like projections are what we are going to go ahead and call as our root hair cells. So when we're going to go ahead and talk about it, now, as you can see from the diagram, if the water has to move into the xylem vessel from the root, it has to go through a certain pathway, right? Water needs to enter through root hair cell. So the root hair cell is going to be responsible for absorbing the water from the soil, right? Then it needs to travel through the cortex before it can reach the xylem vessel, because the xylem vessel is towards the center of the root. So the water needs to cross through all the cells because it's right in the center. So all the cells we have in the middle, the water needs to cross through all of them. So the water has to enter from the root hair, but then it has to pass through all of these cells before it can actually go into the xylem vessel. Now, Generally speaking, the process that water takes is quite simple. Water goes from an area of high water potential to an area of low water potential. That's what generally what happens. Okay. The water potential inside the xylem vessel is lower than the water potential in the root hair cells. Hence, the water moves across the cortex down the water potential gradient. Okay. Now, over here in the cortex, the water can take two possible routes. To cross the cortex, water can take two routes. What are the two routes that water can take? The two routes that water can take is that the water can move from cell wall to cell wall without entering the cytoplasm. Without entering the cytoplasm, the water just goes from one cell wall to another. This type of pathway is known as apoplastic. The second possibility that we have is that water first moves into the cytoplasm or vacuole, whatever. Cytoplasm or vacuole, whatever you want to say, of the cortical cell. So cortical cell is the cells of the cortex, right? By osmosis. So with the help of osmosis, the water first enters into the vacuole, into the cytoplasm, and then travel to the next cell with the help of plasmodesmata, because plasmodesmata is the connection. So it can go from one cell ball, one cell, and enter into the cytoplasm. And then from the cytoplasm, it can go into the cell wall. And then in the cell wall, it can use the plasmodesmata. So from the cytoplasm, then the cell wall, and then the plasmodesmata. This sort of a pathway is known as symplastic path. Right? So this is how it is actually going to be occurring. Now, one last thing that we need to okay, hold on. Anybody has anything to ask? Anybody has any confusion?
No.
Aruj, is everything making sense? Anything that you would like to ask? Okay, then. What we will do is that, so we are going to go ahead and stop it over here for today. And in the next class, now we will go ahead and we will just take a look at how the water moves from the soil into the root hair. We're almost done with the topic of xylem, how the water moves from the soil into the root hair. And then we will take a look at the flowing vessels.